Okay, thank you, Waleed. Thank you for inviting me to the meeting. It's been really enjoyable meeting. Both the science has been very good, and the accommodations and the food also very good. So today I'm going to be talking about small molecules binding to protein-protein interfaces. We've been focused in this area for quite some time at Roche Nutley, and we think we've been able to extract some important principles that are generally applicable to this field and that actually have some practical value. So I hope to share some of those with you today and hope that you find them interesting and hopefully you'll find them useful as well. So I was reading Science magazine on the flight over, and I learned two things. One was that the number of proteins in the cell the consensus is now down to about 20,000 proteins. So I'm going to have to revise my slide. But they're starting to try to map out the interactome in the cell. Just about every protein interacts with at least one other protein. And some of them serve as hubs, and they actually interact with a large number of partners. So the estimates now are on the order of there are something like 600,000 protein-protein interactions happening in the cell. And the second thing I learned from reading Science was that the detection methods tend to miss out on some protein-protein interactions involving membrane proteins. So the 600,000 estimate, if anything, is an underestimate. These interactions are, of course, associated with all kinds of important pathways and processes, many of which are linked to disease states. So it would seem that the protein-protein interaction class would represent a very rich source of targets for drug discovery. But actually, if you look at the mode of action of marketed drugs, you find that almost none of them act by modulating a protein-protein interaction. So the question really is, why is this the case? If it's such a rich potential source of targets, why hasn't it been exploited better? There are a variety of reasons. Most protein-protein interaction surfaces are large and somewhat featureless, so they're devoid of the sort of deep subpockets that you might normally find in a drug target, a more attractive drug target situation. So affinity is achieved by summing up a whole large series of weak interactions that are widely spaced, and the small molecule can't duplicate enough of those to achieve affinity. So small molecules do a lot better if you have some somewhat deeper subpockets and if they're located close to each other. So that blanket characterization is a little bit too broad. It is known that in some cases you have these widely spaced interactions, but only a subset of them really are important for achieving affinity. And if they're located close enough together, you may be able to duplicate that with a small molecule. So these are the so-called hotspots. If you have a hotspot and it's sort of drug-sized, you may be able to do something with a small molecule. So it's hard to sort of predict that a priori. You really need to go on a case-by-case basis and see what's happening. The other problem that occurs is when you do screening. So when you do screening with something like a kinase or a well-behaved target, and you get your sort of Gaussian distribution of affinity results, the high-potent hits are usually legitimate hits. In fact, you can even sort of look at them and say, well, this looks like a kinase inhibitor. It's got a donor-acceptor-donor motif or whatever. In protein-protein interactions, it turns out that the high-affinity hits are often bogus. They're not authentic. They're false positives. And the true hits are buried back in the body of the Gaussian. So very important task in following up screening of a protein-protein interaction is to have some way of authenticating the hits and extracting out the valid hits. I think the reason you have all these false positives is protein-protein interaction sites often are quite hydrophobic, and they can attract sort of weak, nonspecific binding that kind of obscures a lot of the results from things like activity assay screening or SPR, or even 1D ligand observe NMR screening. So we have found that one of the key ways of achieving victory is to apply protein observe NMR to authenticate the hits, or to extract the authentic hits out of this mass of false positives. So the protein observe NMR and X-ray crystallography are the two gold standard techniques for producing absolutely reliable validation of screening hits. Another problem, so success in screening, of course, depends on what is in your screening library. And there are cases where we do a screen and then we do hit validation and we end up with absolutely no good hits at all. And the problem there is that these traditional compound libraries that are based on large corporate collections of molecules, may not necessarily contain PPI type scaffolds, or scaffolds that are more likely to inhibit a protein-protein interaction. Now, selectivity is something I won't talk about today, but proteins, of course, have the opportunity of using anti-binding elements to keep away the non-partners. So they can have a protrusion that will keep away the unwanted partners. And small molecules don't have the size to do the strategy. So you've got 600,000 protein-protein interactions. That just means that 599,999 of them might be off-target things that you don't want to hit. So selectivity is a big issue, but it's hard to deal with at the beginning of a program. So I mentioned that a traditional compound library might not contain PPI-type scaffolds. So what really do we mean? What really do we mean by this? So we've been able to learn from the current success stories. There have been a number of success stories where people have found small molecule modulators of protein-protein interactions. If we look at them collectively, we can try to extract some lessons from these. Before I go on, I want to say that almost all of these success stories are cases where first a peptide was identified, and a peptide was able to replicate the entire partner protein. So I wouldn't say that's an absolute requirement for going forward, but I would feel a lot better attacking a system if there was a peptide ligand already identified. And of course, we acknowledge that some of these systems are going to be so complex they won't be able to be modulated by a small molecule. So having a peptide would allow an alternate pathway toward drug discovery. You could imagine developing the peptide into the drug. Okay, so here's sort of a now classic success story. This is the Nutlin small molecule inhibitor series that binds to MDM2 and inhibits its interaction with p53. So p53, there are peptides of p53 that work just fine as replicates of the intact p53 protein. And these peptides, when they bind, they adopt an alpha helical conformation and they insert 3 hydrophobic side chains into subpockets in MDM2. So there's a phenylalanine 19, tryptophan 23, and leucine 26, which are inserted. And we saw the structure of the Nutlin bound, both by NMR and by X-ray. And a couple things kind of jumped out when you overlay the two structures. One is that the organic scaffold is much more efficient at getting these moieties into the subpockets. So nature has to kind of deal with, in order to achieve rigidity, nature has to use a series of hydrogen bonds, and so has to use this whole backbone to achieve a rigid platform from which these moieties can project, whereas the organic scaffold can do this much more efficiently. The other thing you notice is that the trajectories are completely different. So in the early days of doing this kind of thing, we were very concerned about matching exquisitely the entry vectors of all the different moieties, and it turns out that you can come at it from a completely different direction and still achieve mimicry of the hydrophobic interaction. And another thing that you notice is the chemotypes don't necessarily match up. So you can have aliphatic natural side chains being replicated by aromatic and vice versa. So the good news, or the encouraging news, was there seems to be a lot more leeway in terms of inhibiting protein-protein interactions with small molecules than we had feared in the beginning. So that's the good news. It's still incredibly difficult, though. I would say we're still not at a point where we can efficiently look at a bound peptide structure and design an organic small molecule that mimics it. So our second and third generation inhibitors of MDM2 were designed having already seen the Nutlin bound. And doing that just off the peptide hadn't worked so well. So there is room to improve, I think, in terms of going from peptide to small molecule, just de novo design. So by now there are other MDM2 inhibitors identified by other groups. It's instructive, I think, to do sort of a collective comparison of these. So there are a variety of scaffolds utilized, but if you actually then start to look at what submoieties are used to replicate the amino acid chains, you find a lot of similarity coming back. So, for example, in the tryptophan subpocket, you find these sort of bromo-Phe, chloro-Phe groups. Here's a chloroindole, and they all seem to bind in a sort of very similar way, all pretty much coplanar with the parent tryptophan side chain. Over here in the leucine subpocket, again, we're using halogenated Phe, so bromo-Phe, chloro-Phe. And again, they are coplanar with each other, but kind of perpendicular with respect to the original leucine. The Phe pocket is a little more of a varied affair, so you have more of a variety of groups. Here's an ethoxy group, and here's sort of an iodo Phe coming in, just a kind of Phe edge. So it's a little more varied, but again, it's coming in a little more of a perpendicular orientation than the original Phe is. So there are some similarities, but some differences. But again, a variety of different scaffolds. The scaffolds aren't worrying at all about replicating the backbone. They're just delivering the moieties in the right geometric orientation. So there's a variety of scaffolds that are possible. We have now reported recently a completely new class of MDM2 inhibitor, as shown here. In the initial hit verification stages where we were using the NMR, we noticed this interesting upfield shift of tyrosine 63. This we had only seen before when the peptide was bound, and it was due to shielding by the peptide phenylalanine. So the Phe19 was shielding and causing this shift. And so when we tried to think about how this must dock into the binding site, we figured, well, this sort of indole-looking thing must go where the tryptophan is. Then we need something to go into the Phe pocket to cause this shielding, and we really couldn't orient it to achieve those two things. The other thing that was a little weird was we saw a lot of broadening in the NMR when the compound was introduced. Then we did, our colleagues did a size exclusion column experiment showing that upon addition of the inhibitor MDMX, oh, sorry, I've switched to MDMX, which is a highly homologous protein to MDM2. The MDMX seemed to dimerize upon addition of the small molecule inhibitor. And then X-ray crystallography pretty much explained what was happening. And it turns out that the moiety that was going into the trip pocket was the difluoro-Phe and not the indole. The indole was going into the Phe pocket and causing that shielding that we saw in the NMR. And the interesting thing was that these two binding strategies are being carried out by two separate inhibitor molecules. So the inhibitor molecule dimerizes, and then the protein itself dimerizes. So it's a 2-2 situation. And then key pockets are filled by alternate members of the pair. And then, of course, you can see that there's a lot of stacking, both inhibitor-inhibitor stacking, and then there's some other stacking between this stack and the rest of the protein. So this was a really bizarre but interesting sort of strategy for binding. And we thought about trying to achieve both of these with a single molecule, or maybe dimerizing the inhibitor to come up with a single molecule, but we haven't really, we're not ready to report on anything there. So that shows that here's a completely different strategy to achieve a high affinity inhibitor of this system. Okay, so I think in the interest of time, I won't go through. I was going to go through a whole series of success stories, but I'll kind of just flip through them, and you can hopefully you'll extract a couple notions. One is that a lot of these small molecule inhibitors look quite three-dimensional. And you'll also see that a lot of mimicking of the natural side chains is done by very different chemical moieties. But the positioning is quite close. Okay, so in terms of the three-dimensionality, one trend that we sort of just phenomenologically viewed was that a lot of these small molecule inhibitors are more three-dimensional than typical drugs. So to do a little more rigorous characterization of that notion, we collected all of the cases in the PDB where there is a structure of a marketed drug complex to its target protein. So we found 63 of those. And then we looked at small molecule protein-protein inhibitors where there's an X-ray structure or an NMR structure of the inhibitor bound to the target protein, and there are 25 of those. And we basically compared their three-dimensionality. So we have a program, Mo, in-house that will give you back the three standard dimensions of a ligand. So it's a unitless coordinate system. It basically tells you dimensions 1, 2, 3. And so we then compared the third dimension values. That's basically the thickness of the molecule. So you can see that it's something completely flat is 0, and then something like cubane is quite high. And so what I've done is just plot the standard dimension 3 values for typical drugs and for these protein-protein modulators. And I've done it in a kind of, I don't know, semi-artistic way so that you can visually see that the protein-protein modulators do tend to be more three-dimensional than a typical drug. And that seems to be a pretty strong trend. And you can even do statistics and show that what I just said is statistically valid. So why can't something flat work? I mean, you can kind of intellectually, you can do a thing where you try to just manually dock something flat into the MDM2 pocket, and you see that you have reasonable landing spot for it. It seems to fit okay. But we know that in our high throughput screening, we screened ultimately something like 1 million compounds, and many of them are flat, and none of those really emerged as good hits. All of the good hits, actually there were 2 good hits, and they were both quite three-dimensional. So it seems like something flat just does not do well in this pocket. And the three-dimensionality, as you may have noticed from my quick flip through, in some cases you have a rigid scaffold, and the three-dimensionality is just a consequence of that, so there's no choice. But in other cases, the three-dimensionality comes from stacking. So you have a molecule that could be quite flat, floating around in solution, and when it binds, it stacks. So there are kind of 2 ways to achieve the three-dimensionality. And the value is it imparts rigidity, and rigidity is good because you avoid the problems with trying to get into the active conformation. And when you're rigid, you're able maybe to displace flexible side chains and kind of carve out your own binding subpocket, and we do see that in a number of cases. Basically, whatever nature is offering you, so if the protein is offering you a very rugged three-dimensional terrain, then you've got to adapt to that, and you have to be three-dimensional in order to exploit that properly. Probably don't have time to get into this, but there is a paradox, which I said in the beginning that these protein-protein sites are very shallow. If the inhibitors are very three-dimensional and they're sitting in a shallow pocket, something's got to give. So part of the inhibitor must be sticking out of the pocket. And that is observed quite a bit. So I won't go into this part of it, but that's kind of a observation that we don't have a complete explanation for in terms of what's the value of that. Just a couple of quick observations, anecdotal, but I think informative. So as we do screens of these PPI targets, we find that the hit molecules are statistically more three-dimensional than those from screens involving other target classes. And we've also noticed that during the lead optimization phase of a PPI program, increases in potency are accompanied by increases in three-dimensionality. So three-dimensionality seems to be an important characteristic, and we were asking, can we skew our screening library by adding more three-dimensional compounds to increase our chances of finding PPI hits? So we had a little bit of a campaign where we designed things that were rigid and more three-dimensional, and then seeded the library with these and called it sort of the PPI library. And we found that hits from the PPI, the hit rate of the PPI library against PPI targets was higher than that of the conventional library. And some of these were actually quite interesting. So here's, this is just showing in the case of MDM2 that we have compounds that bind and perturb in the same pocket that the p53 peptide binds into. So we can validate these hits with NMR and show that they're binding in the business pocket. And as I mentioned, in the initial high throughput screen, we really only got 2 good hits from the entire compound corporate library. So skewing things toward three-dimensionality seems to help a little bit. So the final thing I want to talk about real quick is applying a fragment approach to a PPI system. So I mentioned that the PPI success story hits tend to be kind of large. We know that for a typical drug, molecular weight around 500, fragment libraries are usually 150 to 250. But if your final drug is 700, do you need a larger fragment to get anywhere. So what we've asked is, could the Nutlin scaffold have been discovered via a fragment approach? And we've done this by deconstructing the Nutlin. So here's a typical Nutlin. Just schematically, we've kind of turned it into the Lu, Trip, and Phi sub-pocket fillers, and then this piece that sticks out into solvent. And then we've, from synthetic things that came out of the synthesis or things that we've specifically synthesized now to be fragments, we've been able to check a number of molecules representing fragments of the Nutlin. And what we find so far, so this is sort of in progress, but I think it's interesting, and I kind of wanted to share it at this point. If you just have individual sub-pocket fillers or the core, you don't see much binding. If you look at things with 3 legs, or sort of 3 sub-pocket fillers, they all do bind. So having 3 out of 4 is sufficient for binding. If you look at the ones that just have 2 legs, it's a little bit of a mix. So if you've got the Phi and the out piece, you don't bind. If you've got Phi and Trip, you do bind. And if you've got Lu and Trip, you do bind. So there are fragments with just 2 sub-pocket fillers that do bind. Their molecular weight is a little bit on the high side for a fragment. So it's sort of within the fragment zone, but it's way toward the high end. And so, yes, hypothetically, we could have discovered the Nutlin by starting with a fragment and then going to a 3-legged version and then finding the Nutlin. And Abbott has done a similar thing. They've deconstructed their BCL2 inhibitor down to the smallest fragment that binds. And that, again, has a molecular weight of 293, which is, again, toward the high side of the fragment range. So to summarize, the key attributes of successful small molecule modulators of PPI systems, they tend to be larger. They tend to be more three-dimensional. This can be achieved by stacking. All they need to do is effectively mimic the critical interactions by the natural side chains and not so much the backbone. If we collect the success stories, we find some amino acids more commonly than others being duplicated. There's a lot of leeway with respect to chemotypes and with respect to how they enter the subpockets. So this is encouraging, gives us some leeway. They often have parts sticking out into the solvent. We're not sure why exactly. If you're going to do a fragment approach, you might want to skew your fragment library toward the larger molecular weight side. And again, selectivity I didn't talk much about. And let me just finish by thanking all the people who contributed to what I talked about today. A lot of great colleagues at Roche Nutley. And I thank you for sticking around to the bitter end and listening to my talk.
Thank you, Dave, for this very interesting lecture. It's open for discussion. Yes?
So could you explain a little bit more what you mean by three-dimensionality? I mean, do you mean that your basic scaffold that you're putting on your groups shouldn't be planar? Or do you mean rather that the basic groups that you're putting on should have inherently three-dimensionality themselves?
Yeah, so we're saying that the final product viewed in toto, if you measure its 3 dimensions, its third dimension should be rather sizable rather than the whole thing being flat.
I mean, so how can you judge it? Let's say that you have an aromatic group. So an aromatic group can be like this or like that, and it has quite different dimensions when it's like this than when it's like that. And if you put in 3 of them, you know, then the question is, how do you evaluate your three-dimensionality before the fact when you see the interaction?
So in this study, we always used the bound conformation. But you're right. How do you then know what you're aiming for? So especially if you allow stacking, that adds a whole other uncertainty. So it's I'm not saying it's easy to know what to do, or how to do it, but it seems like if you can achieve three-dimensionality at the end, you might do better. So, yeah. But you're right, it's not clear-cut how to get there.
Any other question?
Your statement that only the side chains are relevant if you have a proper scaffold, and it's not the... Does that imply if you made the retro-inverted peptide, native peptide of the p53 sequence, it would still be binding?
It should. Yeah, I mean, it seems like there are some systems where backbone hydrogen bonds are critical, but there are many where they're not. So in those cases, the backbone shouldn't matter. There are MDM2 inhibitors that have a beta hairpin backbone.
Is that a general rule, or if there is possibility of a general rule for PPIs that they should, if it's only the side chains are relevant for binding?
I wouldn't say it's a rule, but it's more of a statistical thing that probably many more than half of them you won't need to duplicate the backbone, I would guess.
Okay, so please joining me to thank all the four speakers of today morning for a great session.